Hi, I'm Jenna Myers, and today we're going to be talking about the determination of gender as it relates to genes versus hormones in that process. So let's go ahead and get started. Pull up my presentation here. Okay, again, like I said, we're going to be talking about comparing genes versus hormones in determination of sex in utero. This is just kind of a intro slide, talks about the different sex organs. So there are three structures throughout this whole process that we're going to be talking about as they develop. Just in general terms, the three structures are gonads, which are they produce gametes, which are the sperm and eggs, and in each gonad, depending on if it develops into male or female. In males, it's testes that produce sperm, and females, it's ovaries that produce eggs. And prior to differentiation, these cells are called germ cells. And the next organ is the internal genitalia, which consist of accessory glands and ducts that connect the internal organs to the external environment. And then you have your external genitalia, which is kind of self-explanatory, which is all of the external reproductive structures. So this is going to be kind of the layout of my presentation, as we're going to talk about each of them as they go along in their differentiation, each of these organs. So keep those in mind. It helped when I was studying, definitely. Okay, so now we're going to talk about chromosomes. In humans, we have 23 pairs, 22 pairs of autosomes, and then one pair of sex chromosomes, and you can see that in this photo in the corner here. You can see each pair, all the 22 autosomes, and then your sex chromosomes. So sexual development is programmed in the human genome, and each nucleated cell in the body has 46 chromosomes. So each of these in pairs, except eggs and sperm. And so these 22 matched chromosomes are called diploid because they're matched. And then haploid chromosomes are these guys, the sex chromosomes. And so we call diploid 2N and haploid 1N. We won't get into that too much, but it's just a good way to remember. And so when it comes to sex chromosomes, you're either X or Y, which you can see here, and then you can see in this table here. And then, like we talked about, eggs and sperm are haploid, so they're 1N. And then we'll go more into sex chromosomes in a minute, but it's just good to have that image there, just to look at. So there is a title in the book, or a subsection called Sex Chromosomes Determine Genetic Sex, which I thought was helpful. So I just kind of copy-pasted it there. Regarding the sex chromosomes, each gender carries an X, and the absence of a Y means development as a female. So I think this chart is really helpful. So a female parent obviously has XX, and then a male has XY, because each gender does have an X chromosome, and it's the Y that determines whether or not the fetus will develop as a male or female. You can see here, female produces 2X eggs, and then sperm X and Y. And so the combination of these are, if you get an XX, female offspring, and then XY, male offspring. It makes sense. I think this chart is really helpful too. And in the book, they also kind of talked about, well, what if you don't get two X's or an XY? What if you get an X and then nothing, which is here, it's called Turner syndrome, and they kind of exhibit male development. Genetically they're female, but they have some male characteristics as they get older. And then the book also talks about Barr bodies, which as a female, you use one of your X chromosomes to determine, to determine which one's going to be active. And so the inactive X chromosome actually becomes a Barr body, which is just a clump of cells that doesn't get used. And then in males, it's XY. Kind of what we talked about, the presence or absence of this Y determines the development. And every living human, I suppose, needs an X chromosome. So in this case, Turner syndrome, X with nothing else will survive, but Y without an X will not survive, which is kind of interesting because the larger X chromosome has some information in it that's required to live, basically. So at Y, nothing is death, which is interesting. So we're going to move on here. So this is kind of a busy slide, but I just kind of talk about sexual differentiation. And I know this is a lot of information, but it's an image in the book that's helpful to look at, so you can kind of see the development of certain things as internal organs, external genitalia at certain timelines in the development for both male and female. So I'm not going to go over this in detail, but it's a helpful tool to look at in the book if you're wanting to study. So in differentiation, the cells don't actually differentiate until 7 weeks of development. And prior to this, gonads and tissues are bipotential, which I wrote up here, which basically means they have, they're undifferentiated and they have the potential and are equipped to become either male or female. So the bipotential gonad, which is one of those sex organs that we talked about in the beginning, contains both an outer cortex and inner medulla. And so depending on if the fetus develops as a male or female, if it's a male, the medulla will become testes, and then if it's a female, the cortex will become ovarian tissue, and then the other tissue will regress. So in a male, medulla will become the testes and the cortex will regress. Female, opposite is true. As far as the bipotential inner genitalia, consists of 2 ducts, the Wolffian ducts and the Mullerian ducts. And the Wolffian ducts will develop in a male, and then the Mullerian ducts will develop in a female, and then each one of those will regress depending on if it's male or female. So a Wolffian duct in a female will regress, Mullerian duct in a male will also regress. And then moving on to the bipotential external genitalia, these all consist of the same thing because they're bipotential, haven't developed yet. They consist of the genital tubercle, urethral folds and grooves, and the labioscrotal swellings. And you can kind of see that here, and they list all of those. So this is the bipotential, and then once they develop down here. But this is what we're talking about in this slide. So this image is helpful just to see that piece of it. And then as development continues, each of these will become specified, and we'll talk about that a little bit next. If I can get my notes here. Okay. So let's say they develop into either male or female. What happens next? So in a male, the SRY gene produces TDF and binds to DNA. Well, what the heck does that mean? Because we haven't talked about that yet. So in a male, they have this thing called the SRY gene, which this is what the actual determination of sex depends on, is the presence or absence of this SRY gene. And this stands for the sex-determining region of the Y chromosome. They kind of talk about that abbreviation in the book. I'm not sure if that's what it actually stands for, but it's a helpful tool. And it makes sense, because there's a Y in it. So if the developing embryo contains this SRY gene, then the human will develop as a male. And this SRY gene produces TDF, which is testes determining factor, and this binds to DNA that activates additional genes that kind of direct the development of the medulla, like we talked about earlier, into the testes. Oh, it's right here. Perfect. And that's what that TDF does. And I know everybody's probably thinking, what about testosterone? Everybody's thinking about that as a male hormone. It's not required. in this stage of the game. And so once the gonads are differentiated, the testes will secrete AMH, which is anti-Mullerian hormone. And remember that the Mullerian ducts in males will regress. So it secretes that hormone, and that's what causes that regression. Also secretes androgens and DHT. So the kind of cells that secrete these are right here. So Sertoli cells are the ones that secrete AMH, and the Leydig cells secrete androgens and DHT. And androgens contain, or testosterone is an androgen, and then DHT is a derivative of testosterone. So in females, there's no SRY gene, no sex-determining region of the Y chromosome, and so that cascades into all these different things. So they're kind of like algorithms. So if there's an SRY present, then it'll defer to its algorithm that kind of goes down the line of all the development. So no SRY gene. The book talks about a complex development of ovaries regarding hormones and genes there, so they don't specify the certain hormones like they do and genes in the male development. So no SRY gene triggers the bipotential cortex to develop into ovarian tissue and for the medulla to reduce or degenerate. And then the Mullerian ducts will develop into vagina, uterus, fallopian tubes in the absence of that anti-Mullerian hormone. The Wolffian ducts will degenerate, and then without DHT, we see here, the external genitalia become female. So essentially, in female development, it's sort of the absence of all of these hormones and genes in the male development. So that's kind of how I remembered that. And in a summary slide, we just kind of have a table of kind of everything we just talked about, the male, and then it gives a timeline. So 6 weeks, 10 weeks birth. And this table is also in the book. I just kind of put it into my own words in my slide. So at 6 weeks, male gonadal cortex will regress, medulla will make its way to testes, and the Wolffian duct produces all of these structures by the time of birth, and we'll reiterate that down here. And then the Mullerian duct regresses. The opposite is true in female. We won't go over that. So basically, I'm just going to go over this, and then basically the opposite is true in a female. And so at 10 weeks is when we see that SRY gene, and this will develop testes from the medulla, and then the anti-Mullerian hormone will cause them to disappear. Opposite is true here. We also talk about the absence of testosterone causes the Wolffian ducts to regress. We already talked about this here. So gonadal cortex to ovaries. Absence of this SRY gene is now under the influence of female-specific genes, which they don't really talk of specifics in the book, but I guess that's the extent of what we need to know for the class. And then at birth, testosterone will form the testes, seminal vesicles, vas deferens, epididymis, and DHT controls the prostate development. And then in the absence of AMH, allows the Mullerian duct to develop in the fallopian tubes, uterus, and vagina. So that's just kind of a nice overview. Thank you for listening to my presentation.